Welcome to Biology 103, Cellular and Organismal Biology. This is a course that's geared towards humanities students. So students that may not continue in biology, but students that hopefully find biology really interesting. So hopefully you're taking this because you do like biology and you want to know a bit more about it. In this course, we're going to examine how life is organized, how your cells are organized, for instance. We'll take a look at the complexity of life. You are a very complex machine. And we'll talk about how the information that's contained within DNA makes all of this possible. DNA is a blueprint. There's a language there, and we'll talk about how that language works. So some of the big questions we'll talk about, we'll start off by talking about how organic molecules, those are molecules that have a backbone of carbon, are used to build cells and how cells are used to build bodies. We'll talk about how DNA works. So it stores information, but how does it do that? You pass DNA down to your offspring and they pass it down to their offspring. How does that work? DNA codes for the diversity of life that we see. We have so many very different organisms on the planet, a few million different species. But in some ways, all those organisms, all those cells are the same. There's some uniformity as well. And we'll talk about how that works, and we'll talk about DNA's role in that process. The Earth is a pretty special place, as I hope you'll agree. There might be life out there somewhere. There's lots of stars out there. There's lots of planets out there. No doubt there could be some life out there. But at the moment, the only life we know exists on the planet Earth. And we're going to start out kind of broad and look at the hierarchy of biology. What I mean by that is we can look at the Earth as kind of a big living thing. It's covered in a layer of living stuff. And then that's broken up into ecosystems and communities, that's broken up into populations, etc. And what we're going to do in this topic is introduce you to that biological hierarchy and also introduce you to the scientific method. That's the process that scientists use to learn stuff. So as a bit of an overview here with biological hierarchies, we'll talk about how life is structured. We'll talk about the levels of order that we see in living things, that we see in our environment, that we see on our planet Earth, and in the universe for that matter. We'll move on, we'll talk about what is life. That might seem like a really easy question, but you might be surprised to learn that biologists struggle with that question. What makes one thing non-living and one thing living? There's more than just one characteristic we need to look at. Then we'll talk about how life adapts over time. So very briefly, we'll look at evolution. And you'll come back to that a little bit in this course and more in Biology 104 if you take that course. We'll talk about the scientific method. We'll talk about how we study the universe, how we study living things, how we study physics, etc. There's a method to our madness here. We'll talk about what the limitations are of science. There's certain things we can't question using the scientific method. Okay, so starting with the hierarchies. This image you're seeing here is an artist representation of what we think our galaxy looks like. This is the Milky Way galaxy. It's pretty big, as you can see by the scale at the bottom there. That's a pretty typical galaxy. Now the Earth, as you know, is one of 8 planets circling around the sun. Sorry, Pluto. And our sun is a star. And that star is just one of 100 billion stars just in our galaxy, just in the Milky Way galaxy. Our galaxy is one of 100 billion galaxies in our universe. And those are mind-boggling numbers. And it shouldn't make you feel insignificant. Should make you feel pretty significant because you are here. You're alive. You're cognitive. You're thinking about this stuff. And yeah, I mean, most of the universe is probably empty, probably devoid of life. Again, eventually we might discover some more life out there, and I'll talk about that a bit later on. But my point here is that a hierarchy consists of a small category inside a larger category inside a larger category. So we have our sun, which is part of the Milky Way galaxy. The Milky Way galaxy is part of the universe, which contains lots of galaxies. And this is a pattern we see over and over again in the universe. So we have planets and stars and big spaces between them. Our solar system is part of a galaxy, but the nearest solar system is very, very far away. And then our galaxy is part of the universe, but the distance between galaxies is enormous. It's mostly empty space. And you might be scratching your head and saying, why am I talking about this in a biology course? Just because it's kind of cool, and I wanted to start off with the broadest stuff. It'll all make sense in a moment. Now, there is a really good video on this. I'm not going to go through this because this is astronomy. You don't need to know any astronomy for this course, so you can ignore all that stuff I just talked about if you want to. But there's a classic video called Powers of 10, which puts things in perspective. Like I said, I'm not going to ask you questions about this, but I'll put a link up on our Moodle page that will link you to this video so you can explore that a bit more if it's something you're interested in. Okay, so let's get down to the biology here. The Earth is surrounded by a thin layer known as the biosphere that can support life. So of course we have life living on the surface of the planet, we have life living in the oceans, we have life living a little bit deeper down into the soil and into the rock. We have some things that are floating around in the air, but this is a pretty thin layer. This is the layer where life is confined, and that's the biosphere. So that's our biggest kind of category here. Now within the biosphere we have lots of different ecosystems. So we have the boreal forest, for instance. We have the tropical forest, the rainforest. We have large Great Lakes and things like that. These are ecosystems. For an ecosystem, we're talking about a smaller part of the biosphere. And we're talking about everything that lives in that area. So all the plants and animals and bacteria, etc. But also with ecosystems, we're including the physical characteristics. The physical properties, the pH, the temperature, the amount of moisture, that kind of thing. So an ecosystem considers the biotic components, biotic means alive, and also the abiotic components. Abiotic means non-living things. Now, if we go a little bit further down, we can look at communities. So within the ecosystem, if we throw out all the physical non-living stuff, we're left with communities. Communities are groups of plants and animals and bacteria, living things that are all kind of interconnected. They're all maybe dependent on each other, maybe they're fighting each other, but they're all interacting with each other. So communities are just the living component of the ecosystem. Going down a bit further, we have populations. A population is a group of individuals of the same species. So in the example we see here, we're looking at a forest ecosystem, so maybe a temperate forest. The community would be all the different tree species. We've got a deer there, we've got mice and insects and bacteria and so on. That would be the community. And now when we get to the population level, maybe we're just looking at one particular species of tree. Maybe we're just looking at oaks, for instance. Then we get to the organismal level. So the population is all members of the same species. Organisms, of course, are separate living things. Now that tree, as we'll see, consists of many, many millions or billions of cells, but we're looking at just one tree when we talk about an organism. So we've looked at the entire organism. That would be an oak tree, for instance, although in this case we've got a maple tree. The organism is made up of different organs and organ systems. An organ is something that's focused on one thing. Your stomach, for instance, is an organ, and it's focused on helping you break down food. Plants have organs as well, and within a plant, the leaf is an organ, the stem is an organ, the root is an organ. Those are all doing very different things. So within this maple leaf, we have several distinct tissues. A tissue is a collection of cells that are all working together to do one thing as well. So within the leaf, we have a tissue on the outside known as the epidermis that helps protect it. And in the middle, we have a tissue known as parenchyma. Don't worry about that at all. But parenchyma is full of cells that are photosynthetic. So the function of the leaf is to trap sunlight, and that energy from the sunlight is going to be used to make food, and that occurs in the parenchyma tissue. Now we can go further. Of course, we're getting in smaller and smaller and smaller, looking at smaller and smaller pieces. Within that parenchyma tissue, we have individual cells. And those cells are filled with chloroplasts. And chloroplasts are an organelle. So within a cell, we have these separate compartments. Organelle literally means little organ. So within your cells, you don't have chloroplasts, but you have mitochondria, you have the Golgi body, you have the endoplasmic reticulum, all this stuff we'll talk about later, but these are compartments within the cell. So we have an organ that's made up of tissues, tissues are made up of cells, cells contain organelles, and then if we go a bit further, of course, the organelles are made up of molecules. And the molecule you're seeing there is actually chlorophyll, which is what's responsible for trapping the light energy. So let's move away from plants and let's look at an animal. Let's look at us. Let's look at humans. And this time we'll do it in the opposite direction. So again, a hierarchy just refers to the fact that you have little bits making up bigger bits, etc. So within your body, you have atoms. And the atoms come together and they make molecules. And the example here is a phospholipid. This is something that's found in the membranes of all of your cells. Those molecules make up organelles, little compartments within the cell, and the organelles together make up the cell itself. Cells clump together and form tissues. So you have tissues within your skin, for instance, that protect you. And these cells are all working together towards that goal. The tissues make up organs. So for instance, we have cells that come together to form your stomach and form your intestines, your small intestine, your large intestine, form your esophagus, that's the tube that the food goes down when you swallow. And all of those organs come together to form an organ system. In that case, we're talking about the digestive system. All of that, all of these organs and organ systems come together to form an organism. So you are an organism. And you make up part of a population. So if you're up there in Prince George, there's a population of humans, right? It's just a collection of individual organisms of the same species, human in this case. That would be a population. And populations come together and they form ecosystems. So if we went out into the woods, we would have deer. We would have a population of deer. We'd have a population of mice. We'd have a population of dragonflies, etc. And all of those would come together to form a community. And then if we throw in the temperature and pH and all that physical stuff, we have an ecosystem. And then ecosystems all clump together to form the biosphere, which is the highest level, the highest category when we're talking about biology. In this course, we will talk about cell biology quite a bit. It's also known as cytology. We'll come back to that. But that's the study of cells and the bits and pieces of cells and how cells work and so on. So within cells, we have, once again, molecules like phospholipids and proteins and carbohydrates, nucleic acids, we'll talk about all that stuff, that come together to form organelles and structures that then come together to form the entire cell. And biology covers a huge range of sizes of things, as we've just talked about. Molecules and atoms, those are tiny. The biosphere is huge. And it's important that we kind of wrap our head around this scale quickly here. What you're seeing here is kind of the range of things, the spectrum of things that a biologist might study. Now let's start with our friendly dog here. So right here on the scale, let's say you got a big dog, it might be about 1 meter in length. Now as we go up towards the right, things are getting bigger and bigger, and as we go towards the left, things are getting smaller and smaller. But notice that the scale is not consistent. It's a logarithmic scale. And what that means is as we go from 1 meter to 10 meters, we've gone up by a factor of 10. As we go from 1 meter to 100 millimeters, we've gone down by a factor of 10. So 100 millimeters is 1/10 of a meter. So as we go towards the left on this diagram, with each one of those markings on the scale, things are getting 10 times smaller. So if we go down to 1 millimeter, so we've got 100 times smaller from a meter, we have some of the largest cells that are found on the planet. There are some that are bigger, but one of the largest cells that's fairly common is an egg cell. So an egg cell in a human or in another mammal. So we do produce egg cells, of course. They're shed every month as humans during the menstrual cycle. That cell is about 1 millimeter across. You could just make it out with your bare eye if you're lucky enough to see one, I guess. But that cell is the largest cell in your body. And then as we get smaller and smaller beyond 1 millimeter, we start to use something known as a micrometer. And it's got kind of a weird symbol. It looks like a U, but with kind of a tail in front of it. That's the Greek letter mu. 1 micrometer is 1/1000th the size of a millimeter. So when we go from that human egg down to a mitochondrion, the mitochondrion is 1/1000th the size of the human egg. Now we can go down further and we can talk about nanometers. And a nanometer is 1/1000th the size of a micrometer. So just to point out here, we're talking about a huge scale of stuff that we're going to look at in this course and that biologists look at. When we get down to atoms, we're talking about things that are incredibly tiny. And as you can see down the bottom of this diagram, some of this is visible with a compound light microscope. That's the kind of microscope you'll have a chance to use in the lab. But these really tiny things, we need very sophisticated, very expensive electron microscopes. All right, so let's move beyond the scale of stuff and let's get into something new here. So the second part here, we're going to look at what actually is life. And as you might imagine, in a biology course, that's a pretty important question. And it's a question that biologists still kind of struggle with. So what you're seeing here are a number of different organisms. We've got, first of all, the human on the Life cereal ad. We've got some bacteria. That's the next photo there. We've got some archaea. I'll talk about what that is in just a moment. A houseplant, some fungi, and a cheetah. Now they all seem very, very different. And they are, but they do share some similarities. All living cells have some things that they need to do in order to survive. And you are, of course, made up of cells. You can think of yourself as a colony of cells. So, of course, you usually think of yourself as one thing, one living thing, but you really are a colony of over 100 trillion cells. And all of those cells have to do certain things to stay alive. So how do we define life? There isn't one single characteristic. And I've come up with several here, and you can probably come up with a few on your own, but I think these are some really good ones here. We know that life reproduces, for instance. That's the second characteristic here. And you might think, okay, that's a pretty good characteristic. You know, maybe you can't think of anything that's not alive that reproduces. But if you think a bit harder, you might come up with some stuff. So for instance, a computer virus. A computer virus uses your hardware, your computer, uses the software that's in that computer, and it reproduces. But I don't think most people would consider a computer virus to be alive. And in fact, most biologists wouldn't consider a biological virus like the coronavirus to be alive either. We'll talk about why that is in just a moment. You might think, okay, growth is a pretty good characteristic of living things. I can't think of anything that's not alive that grows. When you think a bit harder, what about crystals? A crystal will grow over time. Even just an ice crystal will grow over time, and it's obviously not alive. So the point is that living things don't have just one characteristic that defines them. There's a bunch of things that all have to be there in order for us to consider something as living. And like I said, this is the list that I've come up with. I think this is a pretty good overview of what defines life. The first one there is order. So you're highly ordered. You have some very complex molecules in you with very complex structures that have a very precise order to them. And that's something that we see in living organisms. You can reproduce, of course. You grow. So you started out as one single cell, and now you're over 100 trillion cells. Congratulations, by the way. We have energy utilization. So this is sometimes just referred to as metabolism, but the idea is that you take in food, there's energy, there's chemical energy contained within that food, and you're going to release that and you're going to use it to build yourself, to grow, to fix things, to maintain order. We have homeostasis. So inside your body, you maintain a constant temperature, pretty close to 37 degrees Celsius. It varies, it goes up and down a bit, but it tends to stay around there. Your cells maintain homeostasis, they maintain the same salinity and pH, etc. You respond to the environment. If you're standing in the road and a car is coming towards you, hopefully you're going to move out of the way. You're responding to the environment. And also living things adapt over time. So I would say that all life has to have this set of characteristics. Now we live in a pretty exciting time. Within your lifetime, and hopefully within mine, we'll be going to Mars. It's going to happen soon, fingers crossed. And when we get there, it's possible we might find life. There is water on Mars. It's mostly ice, although there's lots of things to suggest that it was liquid in the past. And there may be some simple cells found under the Martian soil. Now, when biologists have a chance to actually look at that, they're going to be looking at those characteristics that we just talked about. So if they find something that has all those characteristics, then yes, that is life. If we manage to go beyond our solar system, we might find life that's very different from what we know, but if it has those characteristics, we can define it as life. So what I'm going to do now is just go through each of those characteristics in a bit more detail. Let's start with order. So what you're seeing on the left is a molecule of DNA. This is a very, very well-ordered molecule. It has to have a very specific shape. So all the little bulges there, those are all atoms that are arranged in a very precise arrangement. And it takes energy to maintain that order. The big thing you're seeing there on the right is a model of the cell membrane, the membrane that surrounds every single one of your cells, also known as the plasma membrane. The red and white things that are bopping around, those are water molecules. And then we've got that yellow, that would be part of a phospholipid. So it's a special fat that helps maintain the structure of the membrane. So the yellow and the green forms a phospholipid. And in the middle, that blue thing, that's a big complex protein, something known as an aquaporin. Again, don't worry about that term, but you can see that the water's going through that quite slowly. That's a protein, it's a channel, it's a protein with big holes in it that allows water to move back and forth. But anyway, everything is always moving around, all the molecules are bopping around, smashing into each other, but within your cells and within your body, we have these highly ordered structures that would break down if we didn't maintain them using quite a lot of energy. The energy used to maintain this comes from your food. So your structures are well ordered, and they would break down if we weren't constantly maintaining them. So all living things do need to reproduce. They need to make copies of themselves, otherwise they'll go extinct. And that can be very complicated or very simple. Towards the left there, we've got a paramecium that has simply elongated, and now it's pinching off into two cells, so it lives as a single cell. So that's a pretty straightforward, very quick way of reproducing. We've got a lion, a lioness here with her cubs. I think we all know how that kind of reproduction works. Pretty straightforward. There's some other animals that are a bit kinky, I guess you could say. They do the same thing, but they make it more complicated. Above the lioness, we have damselflies, which are very much like dragonflies. You've probably seen these before. The one on the top, the blue one there, is a male. The female's underneath. I just find them fascinating because they mate while they're attached to each other. So at the end of the abdomen, the male on the top there has these little pincers, and he grabs onto the neck of the female, and then they fly like this. They actually form this heart-like structure while they mate. I think that's kind of nice. Down the bottom there, and surrounded by black, we have something known as an anglerfish, and they live in the very, very deep ocean. And they're very weird. Again, I don't expect you to know all these details. I just hope you find it interesting. But this big one here is the female, and these little ones hanging on the side are the males. So the males are much, much smaller. These guys live in total darkness. If a male bumps into a female, he grabs onto her, actually bites her. And eventually what happens is the male just merges with the female. He breaks down completely, and all that's left are his testicles. The female gains a pair of testes, and she can do this with more than one male, and then she just keeps basically fertilizing her eggs with her new set of testes. Above that, we have a dandelion. Of course, plants have to reproduce as well. There are sexes in plants. There's male and female plants, or sometimes a plant is male and female, but they do actually produce sperm cells. They're found within pollen, they have sex, and they reproduce new seeds. Growth. Living things grow. So I mentioned before, you started out as a single cell. Your father donated a sperm, your mother donated an egg. Maybe don't think too much about your parents doing that, but the sperm and the egg came together and created a zygote, which is one cell. It divided into 2, divided into 4, divided into 8, etc., until we got to you. And you consist of over 100 trillion cells. So that's some remarkable growth. But even if we're talking about a single-celled organism like an amoeba, when an amoeba splits into 2 cells, the 2 cells, of course, are half as large. They have to grow. So growth is something we always see in living things. At the top there, we can see a chick embryo. developing into a chicken. We can see underneath that, of course, a plant growing, a bean in that case, and then we've got some eggs that have developed into tadpoles, and the tadpoles will develop, in this case, into salamanders. Energy utilization. This is a big one. This covers a lot of stuff. So this is known as metabolism if we talk about the whole set of chemical reactions that happen within your body. The really important chemical reactions are the ones that capture energy that your body can use. Now remember that you're highly ordered. You have large molecules, you have DNA and proteins and carbohydrates, and the structure needs to be maintained, and it takes work and energy to do that. It's just like if you have a house or a car and you don't do anything, it's going to rot, the car's going to rust. You have to put in some energy to maintain that ordered structure. The food that you eat consists of proteins and carbohydrates and lipids, etc. And those are highly ordered large molecules. What your cells do is they break that down into much simpler molecules. So they break the food down into carbon dioxide that you breathe out, and also water. About 10% of the water that you have in your body is actually water you made yourself, which is pretty cool. Well done. And of course, what you're basically doing is burning food with oxygen and then producing these smaller molecules. Now, you may have heard of something called entropy before. Entropy is the idea that things tend to go from an ordered state to a disordered state. When you die, you break down into much smaller molecules like carbon dioxide and water and so on. And that is entropy. It's a tendency towards disorder. The energy that you get from your food, what you're using that for mainly is to fight that breakdown. You're constantly fighting entropy. And of course, when you die, you lose because you don't have that energy source anymore. So you're fighting to maintain order. That requires energy because you're kind of fighting the natural order of things. And that energy comes from your food. And again, the whole set of chemical reactions that occur in the body to allow this to happen is known as metabolism. Next, we have homeostasis. So your enzymes, the proteins that you have in your cells that help you break stuff down and release energy and fight entropy and maintain order, those enzymes like things to be 37 degrees. That's the way that we've evolved. That's what happens in all mammals. Mammals tend to have a body temperature close to 37 degrees, and we want to maintain that. So what you can see here with Homer from The Simpsons, if Homer gets cold, then his blood is going to get cold, and that's going to send a message to his brain. There's a part of the brain known as the hypothalamus. Don't worry about that, but it's basically a little bit on the underside of the brain that is constantly monitoring the temperature of your blood. And if it finds that it's too cold, it's going to send a signal out to your muscles, and it's going to cause them to quiver. It's going to cause you to shiver. And that shivering produces heat, and that heat helps warm you up. This is known as a feedback loop. So you'll keep shivering until you're warm enough, and then that cold signal, that stimulus, is not being sent to the hypothalamus anymore, and you'll stop because hopefully you're comfortable. If you're too warm, if your blood is too hot, that sends a signal out to your sweat glands, and you release sweat, you evaporate the sweat, and that cools you. And there's some other things going on as well, but that's the basic feedback loop that we see here. And this happens in every single cell too. Every single cell has to maintain its salt concentrations, its pH, etc. There's these feedback loops that help bring this under control and maintain homeostasis. So homeo means the same. Stasis also means the same. We're trying to maintain the same condition within your cells and within your body. You respond to your environment. You respond to your surroundings. Your cells respond to their surroundings. If, as just mentioned, your body becomes too hot or too cold, you respond. So response to environment is very important. This is part of homeostasis, but also it might relate to just you feeding yourself or you defending yourself. So let's say that you're, I don't know, a cheetah and you see a yummy antelope. You're going to respond by chasing it. If you are a mouse and you see a hawk, you're going to respond by getting out of its way so you don't get eaten. A couple examples here. We have at the top there some plants that are starving for light. They're in a house that seems to be pretty dimly lit. We can see a window to the right, and they're all moving towards the right. The stems are moving towards the right so they can collect more light. We have a chameleon down the bottom here, and it sees something tasty and it's sticking out its tongue, and hopefully that grasshopper, if you're cheering for the grasshopper, is going to respond by jumping out of the way so it doesn't get eaten. So we need to have some response to stimulus or response to the environment, even if we're talking about something that stays still, like the plant. As you can see on the top there, plants can respond. They can't get up and move, but they can also respond to their environment. And finally, we have long-term adaptation. Species change over time, and if they don't, they go extinct. So basically, how evolution works is within any population, you're going to have some organisms that are a little bit better at surviving, maybe a little bit better at attracting mates, and a little bit better at making lots of babies, lots of offspring. And the ones that are really good at that are going to leave behind more information. They're going to pass on more DNA to the next generation. And the genes that work, so the bits of DNA that help them do this, are going to become more common generation by generation. So basically the successful organisms leave behind more offspring, they inherit those traits, and etc. This continues. So what you're seeing here are different types of fish. We have cartilaginous fish, that would be the sharks and the rays, for instance. We have ray-finned fish and teleos. Teleos are what you usually think of when you think of a fish, something like a perch or a salmon or whatever. We have lungfish, which are really interesting because they have these lung-like structures, and they can actually leave the water and breathe air for short periods of time, or in some cases actually they can actually hibernate underground and breathe air for the entire dry season. And then we have tetrapods. Tetrapods are things with 4 limbs that typically crawl around, although we're tetrapods as well. We might talk about that later. But frogs and other amphibians share a lot in common with fish. And there's lots of evidence to suggest that fish gave rise to amphibians, which gave rise to reptiles, and then reptiles gave rise to birds and mammals. And like I said, unfortunately, we're not going to talk a lot about that in this course. I'd love to, because that's kind of my background. That's really my thing. But you will talk about that in Biology 104 a bit more if you take that course. In any case, things change over time. Species will branch off. So one species might give rise to 2 very different populations, and they go their own way and they change over time. And that's evolutionary adaptation. It's thought that life first appeared about 3.8 billion years ago. That's billion with a B. That's a while ago. And we have 3 distinct types of cells. When we're looking at the classification of organisms, the highest classification we can give is something known as domain. So every living thing fits into one of these 3 categories. The bacteria and the archaea are simpler cells. They appeared first. They do not have a nucleus. That's one of their big defining features. So we refer to the bacteria and the archaea as prokaryotes. Pro means before, karyo means nucleus. They appeared first. They're simpler. They appeared before the evolution of the nucleus. The eukarya have a nucleus, they have well-defined organelles like the Golgi body, like the mitochondria, like the chloroplast in photosynthetic members, etc. The eukarya, again, would include us. We are made up of eukaryotic cells. But all of these different organisms can be traced back to one common ancestor. So there was one common ancestor that appeared 3.8 billion years ago, and that gave rise to the bacteria and the archaea, and the eukarya popped up a bit later, a mere 2 billion years ago. The eukarya are interesting in that they're kind of a mixture of the bacteria and archaea. It's thought that we had archaean cells that engulfed bacteria, and the bacteria actually gave rise to the mitochondria and gave rise to the chloroplasts that we see in algae and plants. Let's compare these different cells. So on the left, we have a really big cell. This is a eukaryotic cell. It's an animal cell. If it was a plant cell, it would have a cell wall around the outside. But this is an animal cell. This one has a really big nucleus. It's not always that big in a cell, but the nucleus is a well-defined compartment that contains the DNA. Outside of that, we have mitochondria in orange, as you can see. We have some other little compartments, things like the Golgi body, the endoplasmic reticulum, etc. But structurally speaking, it's a pretty complex cell. Next to that on the right, we have a typical prokaryotic cell. This could be a bacterial cell, as I've shown, or an archaean cell. They look very similar. And in this case, we don't have those compartments. We don't have a nucleus. We do have DNA, of course. We have to have DNA, but it's just in the center of the cell. It's just all kind of coiled up there. Now, I've also drawn a virus here to scale. So all of this is pretty much to scale. The virus is that little tiny green dot there. A virus is not a cell. All living things, including yourself, are made up of cells. Viruses are not cells. They're simpler than cells. They have DNA or RNA as their genetic material, and then around that they have a coat of protein. So it's like a little capsule that contains the DNA or RNA. And then some of them might have a membrane outside of that. But that's it. There's no organelles. There's no machinery, really. A virus cannot reproduce on its own. It has to infect a cell and trick the cell into making copies of it. Bacteria are all around us. Some of them are good, some of them are bad. The majority are either good or really don't care about us. What you're seeing on the left here is a typical bacterium. This is E. coli. This is something you have in your digestive tract. And it's a good one. It helps you break down stuff. Now what you're seeing on the right is a petri dish that someone has put their hand on. Now I'm a little bit skeptical of this. I think they've actually just spread some bacteria on there, but these are bacteria colonies growing on a petri plate. You can see lots of different colors, lots of different shapes and textures. These are all different colonies of bacteria. A colony is just a big clump that contains millions of bacteria. So bacteria settle out on the plate, they divide over and over and over again, and if you incubate the bacteria at 37 degrees for a couple days, you can see something like this, and you might have a chance to do something like that in the lab. So bacteria, you know, they help us make cheese, they spoil food, they cause us illness, they recycle nutrients in the environment, they do all sorts of really relevant and important things. Archaea, on the surface, look very much like bacteria. So what you're seeing in the lower left is what they look like under the microscope, and they look like bacterial cells. They have that prokaryotic structure. They don't have organelles, they don't have a nucleus. But their chemistry, their metabolism is a bit different. It's actually very close to what we see in eukaryotes. What's really interesting about archaea, I think, is that most of them live in really extreme environments. You're never going to get an infection from archaea. You're probably not going to come across it in your house or in your grocery store. They live on the edge. They live on the fringe. And the reason for that is, once again, bacteria and archaea appeared a long time ago, at about the same time. And bacteria proved to be a whole lot more successful. They were a whole lot more better suited to competing. And the archaea responded by specializing in living in really weird places, really extreme places. There's some that live miles under the earth. There's some that actually live in the atmosphere. They just float around. And there's some that live in hot water geysers. And that's what you're seeing here. So this is from Yellowstone, these pictures. And in Yellowstone Park in the US, which I've had the opportunity to visit, it's really amazing. If you have a chance, you should go there. There are many, many hot water pools. There's water that comes out that is very close to boiling. And these prokaryotes can actually live there. And the different colors that you're seeing are different species of archaea. So looking at the image to the right, it's a little bit hotter in the middle. We've got a species there that has kind of a green color. As we go out, as the water comes up and it cools a bit, we have other species that are orange and red and so on that can't tolerate boiling, but they can tolerate 90 degrees. Anyway, archaea have the same structure as bacteria. They're not pathogenic. They're not things you run into very often, except usually in very extreme conditions. Now, the one reason I find them really interesting is that if we do find life on Mars, it'll probably be something very similar to archaea, or it might actually be archaea. Archaea produce spores that can actually survive exposure to radiation, exposure to vacuums. It's possible that spores actually may have spread between planets, but that's kind of beyond this course. Okay, so let's take a quick look at the structure of these cells. This is a typical bacterial cell, and again, archaea would have a similar structure. But we don't have a nucleus. We have a chromosome. The chromosome consists of one massive loop of DNA that's all coiled up in the middle. Imagine if you took a big elastic band and just twisted it and twisted it and twisted it, you'd get this big coil. So it's one single loop of DNA that's sitting in the middle of the cell. Now, they may also have what are called plasmids. These are very small loops of DNA. This is not to scale. They'd be much smaller than that contain just a few genes. Genes, as we'll talk about, are bits of DNA that code for proteins. So the chromosome contains most of the information, contains a couple thousand genes, and then the plasmid contains a few extra genes as well. We have ribosomes mixed in there, but notice that we don't have mitochondria, we don't have a Golgi body, we don't have endoplasmic reticulum, etc. Around that, we have a plasma membrane, and we may have something known as a capsule or slime layer, and this is a thick layer of protein goo that helps protect the cell. We also have a cell wall, so between the capsule and the plasma membrane, we have a cell wall. And in bacteria, the cell wall is made of something called peptidoglycan, which you can ignore for the moment. We might talk about that a bit later. But this is a rigid structure that helps protect the cell. Let's compare that to a eukaryotic cell. So again, eu means true, karyotic means nucleus. We have a true nucleus. We have a compartment dedicated to storing DNA. We don't see that in the bacteria and archaea. So in the middle there in purple, that is the nucleus. And in the very center is the nucleolus. We'll come back to that. On the left, we have a typical animal cell. So we have the nucleus. It's surrounded by this blue structure in this diagram, which is the endoplasmic reticulum. We have the mitochondria, we have the Golgi body, etc. This particular animal cell has a flagellum. That's the little tail that's sticking out that helps it move around. So think of what you have on a sperm cell. You have a flagellum. On the right, we have a eukaryotic cell, but this time it's a plant cell. Plant cells are similar to animal cells, except they have a few additional things. We tend to think of plants as being simpler, but the cells really are a bit more complicated. So in addition to the mitochondria, we have chloroplasts, which capture light energy from the sun to make food, and the mitochondria break that down. We have also a very, very rigid cell wall that surrounds the cell. And that gives the plant support, because of course plants don't have a skeleton. Their skeleton consists of these rigid cell walls. In the middle, you can also see a vacuole, and maybe I'll just highlight that. Again, don't worry about this too much just yet. We'll come back to that. But that's a structure that's used to store water. Plants are happiest if that vacuole is full. So if that vacuole is full of water, it's like a great big balloon, it pushes out against the cell wall and maintains the rigidity of the cell. It maintains the structure of the cell. The main point of this little discussion here, just this brief introduction, is that all life can be traced back to one common ancestor, the universal ancestor. The early cells that appeared 3.8 billion years ago gave rise to the bacteria and the archaea. Incidentally, archae means old. Think of archaeology, the study of old stuff. So these are old cells, the bacteria and the archaea. They've been around a very long time. The eukarya came later, but they're an offshoot of the archaea. It's thought that archaean cells actually engulfed bacteria and didn't break them down. Instead, they kind of enslaved them. And the bacteria that they ingested but couldn't break down became mitochondria. And it's not something we'll talk about too much in this course, but it's something I invite you to look into. It's really quite fascinating. It's known as the endosymbiotic theory. It's something you'll talk about more in 104. And similarly, the plants and the algae that are photosynthetic, they came from archaean cells that engulfed photosynthetic bacteria, and those bacteria gave rise to chloroplasts. But again, that's a bit more detail than I expect you to know. Just realize we have these three broad groups, these three domains of living things. And the eukarya are kind of a mix of bacteria and archaea. We have these two cells that came together to form a new thing, the eukarya. And then the eukarya started to clump together to form multicellular organisms like fungi, animals, and plants. Now, fungi, animals, and plants you're probably familiar with. Everything else in the eukarya is grouped into something known as the protista. Again, we'll come back to that later. The protista are just eukaryotes that basically aren't fungi, animals, or plants. And before we leave this, I'll talk very briefly about viruses. I know I'm throwing a lot at you here, and we'll come back to this and talk about this in more detail. But viruses are not cells. All living things are made up of cells. This is something known as the cell theory. It's a pretty simple theory. It's just that all living things are made up of cells, and cells come from other cells. So cells divide and they give rise to new cells. But viruses are not cells. And that means that for most biologists, they would not be considered as living things. Instead, they're packets of information. That's the coronavirus there on the left. And what you're seeing on the right is a simplified diagram of an enveloped virus like the coronavirus. And I'll explain what that means. So in the middle, we have genetic material. In cells, that's DNA. In viruses, it can be either DNA or RNA. We'll talk about what RNA is in a few topics. Around the genetic material, the information, we have a coat of protein. And then in this particular case, around that, we have an envelope, which is a membrane. Now, the virus didn't make that membrane itself. When it left the cell it was infecting, it stole a chunk of the membrane. It wrapped the membrane around itself and pulled it off. We have some proteins around the outside shown as arrows and diamonds here. And those proteins, the spikes that we see on the coronavirus, are used to help it stick to another cell and get into that cell. But breaking this down, what we have is just a package of information and some proteins that are really sticky that allow it to enter another cell and inject that information. Now, the tricky part is when the information gets into that cell, the cell doesn't realize that that information is not its own information, and it treats it as if it was. So if a virus injects DNA into a cell, the cell says, oh, okay, I need to decode this. And it just willy-nilly decodes that information. It decodes the information in the DNA, uses it to make proteins, and unwittingly manufactures virus. And then the virus leaves. And in the process, it usually destroys the cell or hurts the cell. So viruses will hijack cells. They're a lot like computer viruses. They will take over the hardware and the software of the cell and trick the cell into making copies of them, and in the process they will damage the cell. But again, we'll talk about that in more detail. They're not considered to be alive because they don't have a metabolism. They can't do anything on their own. They don't have the enzymes they need to replicate. They can't divide on their own. They can't really do anything on their own. And when a virus is outside of a cell, it's just sitting there. It's inert. It's inactive. It's not doing anything. Very quickly, we'll take a look at evolution. There's a very famous saying in biology that nothing in biology makes sense except in the light of evolution. And this was made by Dobzhansky, a Russian population geneticist. Don't worry too much about that. But basically what he was calling attention to is that there's many things in living cells and living organisms that kind of don't make sense. They're inefficient. They don't work well. But they make sense if you look at the history of the organism. And what you're seeing here is an example of that. So this is an image that I took during my PhD research that's taking a look at snake evolution. So what you're seeing here is a snake embryo. It's a python, a carpet python if you're into snakes. And we can see some little nubs back here. There's two little nubs at the back of the snake. And those are limb buds. So snakes evolved from organisms that had arms and legs. So they evolved from lizards. And what we're seeing here are these two little limb buds that would normally develop into back legs. Now in pythons and boas, they develop into little spurs. And if you have been around snakes, you might know about this. At the back of boas and pythons, there's these tiny little spurs. And if you look at them closely, they actually do contain some bones. They contain a femur and they contain some of the other bones that we associate with hind limbs. You've probably seen this guy before. This is Charles Darwin. And although he wasn't the first person to think about this stuff, he gave us our current understanding of evolution. And of course, we've expanded on that quite a bit. And well, that was a big deal. We'll talk later about why this theory of evolution was such a big deal, but basically Darwin laid out how things might change over time. He discussed why things might change and how that could happen. He came up with a concept known as natural selection, which has proven to be rather important. One of the things that Darwin considered in the book that he wrote, The Origin of Species, was the fact that humans have changed organisms a lot just through selective or artificial breeding. So for instance, we have lots of different breeds of cats, lots of different breeds of dogs and pigeons and so on. And what happened is we started out with just a wolf, in the case of dogs, and we bred for certain characteristics. So maybe you wanted a dog that had kind of a flat face because it looks more human. Well, what you would do is you would select dogs that had a flat face, a male and a female, and breed them. And they'd pass those traits on to their offspring, and you'd end up with, hopefully, a dog that had a flat face. And then you again would look for individuals that had this kind of weird trait, this weird mutation, this really flat face, and you'd breed those. And through the generations, you could very quickly get something like a bulldog or a pug, something with a very flat face. In terms of crops, we have manipulated our crops a lot by selectively breeding them. Now, what might be really quite astonishing to you is the fact that a lot of crops that we eat that look very, very different are actually all related to each other. So broccoli, cauliflower, cabbage, Brussels sprouts, kale, kohlrabi, they all come from wild mustard, which doesn't look anything like any of those. So in the case of cabbage, for instance, what people did is they took plants that were very, very short to the ground, where all the leaves came out very, very close together. They took those weird mutants and they bred them with other weird mutants that looked the same, and they did that generation after generation to accentuate those traits, and they eventually came up with a cabbage where all the leaves are really stuck close together, so the stem is very, very short. In the case of broccoli, we've got a plant here that has hundreds or thousands of tiny little flowers. If you take a close look at broccoli, there's tiny little flower buds there, and we eat them before they open, but that's a really unusual plant. In the case of cauliflower, we've got broccoli basically that also doesn't have any chlorophyll in it. That's a really weird mutation, but it's something that people have selected for over hundreds of years. So we've looked for things that are very unusual, purposefully bred them together, and come up with something very, very different. That can happen really quickly if we're talking about artificial selection. Really, none of the stuff we eat looks like anything in nature, and that's not due to genetic engineering or anything like that. We'll talk about that stuff at the end of the course. It's due to the fact that we've taken really weird individuals that we've found interesting and bred them together to get really weird offspring, and we've done that through several generations. But this also applies to natural populations. If we have organisms out there, and maybe some of them are a bit unusual, but it's not necessarily a bad thing because it helps them survive, they will be selected for by nature. So in the case of artificial selection, we're picking out the weirdos and we're mating them together and creating the next generation of weirdos and accentuating that each time. In the case of evolution, we have some individuals that are slightly more fit. And what I mean by that is they fit their environment better. They're more likely to survive. Maybe if we're talking about a gazelle or a deer, maybe they're a tiny bit faster they're more likely to get away from predators. Maybe if we're talking about a snake, maybe they're better camouflaged. They can hide and they can ambush their prey. But within a population, we're going to have variation. I mean, just look at the population of humans around you. We all look quite different. And there's these variations. Some of them might be helpful, some of them might not be. And if they are helpful and they make you more likely to survive and they make you better able to produce offspring, also if they make you more attractive to the opposite sex so you can leave more offspring, if you have these traits that make it more likely that you're going to leave behind children, then the traits that made you successful are going to be passed on to your offspring, and those traits are going to become more common in the next generation. So with Darwin, he wrote a book called On the Origin of Species, and just very briefly, I would like to talk about this a lot more. He had these 5 key insights. He noticed that populations overproduce. So if you go out and you look at, let's say, some mice or some robins or some sparrows that you have living on your property, what you might notice is that they produce a lot of offspring. So maybe you've got 2 sparrows, a male and a female, but over the course of a summer, they might produce 10 offspring. They might lay 10 eggs. Not all of those are going to survive. So what they're doing is they're kind of producing all of these different individuals in the hopes that some of them will survive. And the ones that survive are, in all likelihood, the ones that are a little bit more fit. They're more fit to their environment. Populations tend to remain stable in size. So for instance, even though a pair of sparrows might produce 10 babies in 1 year, sparrows aren't exploding and taking over the planet. Instead, they're competing with each other. So what tends to happen in nature is that a pair of mating individuals will produce a lot of offspring. They compete with each other. They're trying to get away from predators. They're trying to get food. There's limited resources. They can't all survive. And only the ones that are best able to get food, find mates, get away from predators, those are the only ones that are going to survive each generation. Again, resources in the environment are limited, so there's not enough food for all the babies that are being produced. I'm not talking about humans specifically here. Humans have kind of changed the game. We'll talk about that later. But for most organisms living in nature, living in the wild, there's not enough resources. All of the young that are produced each generation cannot survive. Individuals vary within a population. So even if we went out and we looked at a population of sparrows, they'd all be slightly different. Their beaks would be slightly different lengths, slightly different widths, slightly different powers. Some of them could chomp on heavy seeds, some not so much. Within a population, there's a lot of variation. And it's a good thing. If you're producing a lot of offspring, you want some variation because the environment changes. And maybe one individual will be successful one particular year, maybe the next year. The climate changes a little bit. The population of predators changes a little bit, and they need to adapt a new strategy. The variation is inherited. So within a population, the traits that we see can be passed down to the next generation. And this is kind of a very, very quick and dirty summary of how evolution works. So individuals compete for resources and mates because there's not enough. They have to kind of fight for it a bit. And traits which increase the chance of survival and the chance of finding a mate and making babies will become more common in the next generation. And like I said, I would love to go into that in a lot more detail, but unfortunately that's not really the focus of this course. And finally, our last part for this introductory lecture, we're going to talk about the scientific method. This is the methodology that scientists use to answer questions, basically. What is science? Well, getting down to the nitty-gritty, it's from the Latin, scientia, which means to know. Don't worry about the Latin, but basically it's a way of understanding things. It's not really a thing. It's a method. It's a lifestyle. It's a way of trying to figure things out. So it's a logical and systematic search for information. And what I mean by systematic is we come up with an idea, we test it. If that doesn't work, we come up with a new idea and we test that. So explanations are constantly changing. Not because we don't know anything, although we don't. I mean, that's one thing that any scientist will tell you. There's so much that we don't know. But we constantly look at things, come up with a hypothesis, which is our tentative explanation. And if that doesn't work, we change the explanation and then we test that. But we keep going and keep going until we come up with something that makes sense and consistently stands up to testing. So we're focusing on very specific questions, as we'll see. So in its purest form, it's the process of organizing empirical data or evidence into a body of knowledge. Now, what do I mean by empirical? Empirical is something you can measure. So I can take a ruler and I can measure something, and I can tell you it's 15 centimeters. That's an empirical bit of data. I've actually gone out and measured it. I can measure the pH of something and tell you it's 7.5. That's an empirical bit of data. Ideally, we want to use empiricism. We want to go out and physically measure stuff. We can't always do that. And if we can't, then we talk about theoretical science. But still, theoretical science shouldn't just come out of nowhere. It should be based on empirical evidence. So how do we do this? Well, scientists are very curious. And any scientist will tell you what gets them excited is what they don't know. I mean, let's face it, if we knew everything, science would stop. There'd be no point to science. There's nothing left to figure out. So scientists are constantly trying to figure out stuff, and scientists more than anyone are very aware of the fact that we don't know a whole lot, and we're trying to learn more. So a good scientist is going to be curious. They're going to look at the world around them and they're going to go, that's odd. How does that work? Let's try to figure that out. And the first step would be to generate a question. So you're looking at stuff, you're curious about it, it generates a question in your mind that you're fascinated by, and then you try to figure out how can I explain that? And to do that, you form a hypothesis. So a hypothesis is kind of a best guess as to what you think is going on. And then, if you can, you devise an experiment that's going to test that. And this is really important, and this is something that a lot of students might miss, and unfortunately some scientists miss. When you design an experiment, you're trying to disprove your hypothesis. It's very difficult to prove something. In fact, that's a word I don't take lightly. I don't use the word proof very often. It's very difficult to prove something beyond any doubt. It is fairly easy to disprove something. So you're trying to disprove your hypothesis, and every time you fail to disprove it, well, that adds some weight to your hypothesis. It adds a little oomph to your idea. So what you do is you formulate a conclusion based on your hypothesis, and then you see if that pans out in your experiment. is your hypothesis supported? And then if you're doing science right, you do this over and over again. It's not enough to do an experiment once. You have to do it many, many times. And we'll go through some examples of all of this in just a moment. But just coming back to that hypothesis, a hypothesis is a hypothetical explanation. You see something out there that's interesting, you're not sure why it happens, but you want to come up with an explanation. It's a best guess, and then you're going to test that. So a tentative answer, tentative meaning that you think it's right, but you're not sure yet. So you have to come up with a good question, and then you have to figure out a way to test it. And your hypothesis is your explanation for what's happening, and it's on trial. You're trying to disprove it. You're trying to show that it's wrong. You come up with your explanation, you think of every experiment you can that will show it to be wrong. And every time you fail to show it's wrong, that adds some weight to your hypothesis. And of course, if you show that it's wrong, then you have to throw out your hypothesis. Your hypothesis has to be testable, and it has to be falsifiable. So what does that mean? This is really important here. I could say that, let's say I pick up a brick and I drop it, and it falls to the ground. I could come up with a whole bunch of hypotheses. I could say that there's invisible fairies that grab onto the brick and they always pull it to the ground. How would I test that? Because they're invisible. I can't test that. So that's a hypothesis that's kind of going nowhere, because there's no way for me to test for the presence of invisible fairies. There's no way to falsify that, because I could set up cameras and things like that, but they're invisible fairies. I can't falsify that hypothesis. If someone said, yeah, there's invisible fairies, there's no way to see them, there's no way to test for them, that's not within the realm of science. I can't test for invisible fairies. So it has to be testable, it has to be falsifiable. So instead, maybe I drop the brick and it falls to the ground, and I say, okay, there's a force. I don't know what that force is, but there's some force that pulls it towards the center of the earth. Maybe that's my hypothesis. So the brick is falling closer to the center of the earth. And I drop it, and okay, seems to pan out. And then I go to China, I go to Russia, I go all around the world, and I drop my brick, and it always falls towards the center of the earth. I haven't shown a whole lot there, but it does fit my hypothesis. My hypothesis suggests that there's a force pulling things to the center of the earth, and I can go all around the earth, which of course is a globe, and I can find that that is true everywhere. That's a start. That is a decent hypothesis. Of course, we have to move beyond that and figure out exactly what gravity is by doing more tests and coming up with more specific hypotheses. Another really important point in science is that we want to keep things simple. And this is sometimes described as Occam's razor. Occam was a monk back in the Middle Ages. He's very interested in studying the world around him. And he came up with this saying that we should always begin with the simplest answer. That's not quite what he said. I'm paraphrasing here. But what we want to do is we want to come up with a very simple hypothesis and test that first. The simplest hypothesis we can think of. And if that hypothesis fails, then we might have to make it a bit more complicated, a bit more convoluted. So hypotheses and theories should be based on what's known as the principle of parsimony. And this is just a really fancy way of saying keep it simple. So the principle of economy or simplicity. So again, going back to my kind of lame example of dropping a brick, I could come up with any number of hypotheses as to why the brick falls down. towards the ground. And those could be really convoluted. But the simplest explanation is there's just a force that pulls it to the center of the Earth. That's the simplest hypothesis we could come up with. We should test that first. If that doesn't work, maybe we'll go back and look at the other suggestions that people might have offered. So the scientific process is where we gather observations, we come up with a question, we come up with a hypothesis, we come up with a prediction based on that, and we test it. So again, we go out into the world, we see something interesting, we come up with a question. Why is this happening? I don't understand this thing. Then we come up with a hypothesis, and it's like, this is my simplest explanation of what's going on. And then with that explanation, we think to ourselves, okay, if my hypothesis is right, what will happen if I do such and such? And then we test that with an experiment. And if our prediction holds out, if we see what we expected, then that supports our hypothesis. If it doesn't, then we have to change our hypothesis. We have to go back and think, okay, well, that didn't work. What else can I come up with? So science is a method where we come up with ideas and we test them. And the more we know about something, the more observations we have, the better our hypothesis is going to be. So the more information we know about our topic, the more specific our hypotheses will be and the better our predictions will be. When it comes to collecting data, we have two types of data. We have what's called quantitative data, and that's something you can put a number on. So I could go outside and I could measure the temperature and I could tell you it's 25 degrees Celsius. I could measure the pH of a solution and tell you it's 6.5. So quantitative is something that we have an absolute number for. We also have qualitative data. Qualitative data is recorded descriptions. So you've witnessed something that's interesting and you've jotted that down in your notebook, or it might be a diagram or a photo. Okay, so we're almost ready to look at some examples of experiments. Before we do, let's just talk about the parts of an experiment. So we always have an experimental variable. This is what we're changing. This is what we're manipulating. So if we're interested in whether or not temperature affects the rate at which a plant grows, for instance, temperature would be the experimental variable. That's the one thing we're going to change. We're going to keep everything else the same, the amount of light that the plants get, the humidity, the amount of water, etc. All that's going to stay the same, but the experimental variable is what we're changing. The experimental group is the set of organisms that we're manipulating, that we're changing this variable for. And then we might have a control group. So maybe I know that this particular plant grows really well at 25 degrees, and I'm going to get some other plants and change the temperature for them and compare them back to this control that I have not manipulated. In that case, that would be a negative control. Now we can also have a positive control, but let's wait on that a bit. Let's wrap our heads around the negative control first, and we'll come back to the positive control. Okay, so here's an example of an experiment you might do. Let's imagine that you're a gardener and every year you grow sunflowers. And what you want more than anything is to grow bigger sunflowers the next year. So you want tall sunflowers. What should you plant them in? Well, you could do an experiment to find out. So for your experiment, you have to have an independent variable. That's also known as the manipulated variable. That's what you're going to change. And we've decided here to grow them in different things. So the independent variable is what we're going to add to the soil. Maybe we're going to add sand, maybe we're going to add some manure, maybe we're going to add some peat, etc. The dependent variable is what changes. We want to measure the height of the sunflowers. So we might take a bunch of sunflowers. Some will just grow in regular potting soil. That's what we've always used. Some will grow in potting soil with sand added, some in potting soil with manure added, etc. And let's say after 3 months, we're going to measure the height. The dependent variable would be the height. It's something we can measure, and it's dependent, hopefully, on the independent variable. So hopefully what we added to the soil makes a difference in the height that we measure. Our experimental group would be the sunflowers we've manipulated. So we have our sunflowers that we're growing in regular potting soil, but then we have our experimental group that we've added different things to. Our negative control would just be the sunflowers that are growing in regular soil, in potting soil. Something that we were used to, something that we've made observations of before. Now, a positive control is something we would only do usually if all the plants come out the exact same height. And I'm going to come back to that. So put that one on hold for a moment, put a pin in that. Our hypothesis might be that you think if you add more organic material to the soil, you'll get better growth. And if that's your hypothesis, then perhaps the plants that you've added manure to, which has a lot of organics, will grow taller. So your hypothesis is that organic material helps the plants grow taller. Your prediction is that the plants that grow with manure added are going to grow the tallest. All right, so again, let's say that we take our seeds, we put them into pots, and we let them grow for 3 months. And this is what we see. This is our end result. And we can measure the height of the plants that result. So the measurement of height would be our dependent variable. The independent variable is, again, what they were grown in. So we have one that was just grown in regular potting soil, and that's going to be our negative control. We're going to use that for comparison. We have one plant that was grown in soil plus sand, another that was grown in soil plus peat, another in soil plus manure, and another one in soil plus gravel. And after our 3 months, we can see that the plant that included manure in its substrate, in its soil, did a lot better. So that seems to support our hypothesis. Now, of course, we'd want to test this several times. The more times we test it, the more sure we are of our hypothesis, the more support we have for our hypothesis. But what if we got this? What if after 3 months, all the plants came out looking exactly the same? We would refer to this as a negative result. And what I mean by that is there was no change. There was no change in the dependent variable. That might be accurate. Maybe these plants just don't care what they grow in. They always come out the same size. Maybe we got a batch of dwarf plants. They can't grow any bigger. Maybe something went wrong though. Maybe there was a really cold night in the greenhouse and that stunted the growth. So when everything comes out the same like this, we're not sure if the experiment worked, because we didn't see any change in the dependent variable, what we're trying to measure. So in a case like this, it's a good idea to go back and do a positive control in addition to your negative control. A positive control is where you do something that you know should bring about a change. So for most plants, if we add fertilizer, they're going to get taller. And that's what we might do. We might do a positive control where we have soil and then we add some fertilizer. We add some nitrogen, which most plants are going to gobble up and use to get much larger. And if we did that and we saw that the plant did get larger, then that would tell us that, okay, these plants are capable of growing taller. So our results are valid. They can grow to different heights. And what we saw is actually valid. Apparently, they don't care about sand, peat, soil, and gravel. So a positive control is where you're testing to see if the independent variable that you're measuring is actually capable of changing. So to summarize, For our little experiment here, again, our problem was, how do we get taller sunflowers? Our question is, what should we plant them in? Our experiment had an independent variable, that was whether or not we added anything to the soil and what we added to the soil. We had a dependent variable, that was what we measured. That was the height of the sunflowers, and hopefully that differed based on the independent variable. We had the experimental group, and that's the plants that were grown with different additives. We had the negative control, and that was just straight up soil. We have our positive control, which was soil with fertilizer. Again, we're just trying to prove that yes, they can vary in height. Our hypothesis might be that organic compounds, so things that come from living organisms like manure, help stuff grow. And our prediction based on that hypothesis would be that manure should have the biggest benefit. Those plants should grow tallest. All right, let's look at another example, this time from mammals like ourselves. Back in the 1800s, the early 1800s, no one knew what the pancreas did. So of course, anatomists had been cutting people and animals open for a long time. They knew about the pancreas and where it was, but they really didn't know what it did. And that was the case until the late 1800s. So there were two Russian researchers. Don't worry about the names or dates. This isn't a history course. But they noticed that dogs that had their pancreas removed, their kennels that they lived in attracted a lot of flies. So this is something that's not uncommon in biology. If you want to figure out what something does, remove it and see what goes wrong. It's not done so much with organs anymore, but if you don't know what a gene does, what you can do is you can disable that gene and then see what goes wrong with the organism. That's basically what these guys were doing. They were taking dogs, they were cutting out their pancreas, and then watching them to see what went wrong in order to figure out what the pancreas does. They were very observant, and they noticed that the kennels of the dogs that didn't have a pancreas attracted a lot of flies. And they came up with the hypothesis that maybe the pancreas regulates sugar levels in the blood and in the urine. And the reason they came to that conclusion is that they felt that the urine that was released by the dogs without a pancreas was sweeter and attracted more flies to their kennels. So they went back and they did a proper experiment to show if that was the case. So their question was pretty straightforward. What they wanted to know was, does the pancreas play a role in regulating the amount of sugar that gets into the urine? And they had a fairly simple, although somewhat brutal, experiment to test this. So you can see there we have the pancreas, and it sits just below the liver. It sits in the first fold of the small intestine, the duodenum. Their independent variable for this experiment was whether or not the pancreas was present. They basically just had two groups of dogs, one that kept their pancreas and one that had it removed. Their dependent variable was measuring the amount of sugar in the urine that was produced. So they're going to go back, do their experiment properly, and they're going to actually measure the amount of sugar that's found in the urine created by dogs that have their pancreas and dogs that have lost their pancreas. The experimental group would be the dogs that have had their pancreas surgically removed. The negative control would be the intact dogs that still have their pancreas, lucky dogs. The positive control, we'll come to that in a second, but in this case, it's a control that's being used to test the reagent that they use to test for sugar. That'll make more sense in a moment. Their hypothesis is that the pancreas does somehow regulate sugar levels. So having a pancreas makes sure that you don't have too much sugar in your body, in your blood, and in your urine. And their prediction based on that is that the dogs that have lost their pancreas should make urine that's sweeter, that has more sugar. So let's take a look at their experiment in a bit more detail here. So we have a dog at the top there. You're seeing the abdomen and the location of the pancreas. You can see that the pancreas sits just below the stomach in the first fold of the intestine. Now they had these dogs and they treated the dogs all the same. They got the same diet, they got the same amount of water, they were kept in the same conditions, same temperature, etc. But one group of dogs had their pancreas removed. That was the experimental group. And the control group, the negative control group, did not have their pancreas removed. Now it's possible that maybe the stress of the surgery changes the amount of sugar in the urine. So even the dogs that didn't have their pancreas removed, they went through the whole process. They were anesthetized, they were cut open, the pancreas was exposed, it wasn't removed, they were stitched back up. So the only difference between the two groups of dogs is that one group had the pancreas removed, the other group did not, but they both had all those surgical steps just to keep everything exactly the same. So they removed the pancreas using a scalpel, not using Photoshop like I've done. And what they did after a couple weeks is they collected urine from the two groups of dogs. Now there's a very simple test for the presence of sugar in urine. You use something called Benedict's reagent, and that's what you see this guy doing here. You add a few drops of this, and it changes color depending on how much sugar is present. Now there's one problem though that we have to take into account, and that's that Benedict's reagent has a shelf life. If you keep it too long, it doesn't work. If you get a bad batch, it might not work. So with Benedict's reagent, when you add it to urine or any kind of solution that has sugar in it, it will change color. If there's a lot of sugar, it'll change red. If there's a little bit, it'll change green. In between, it'll be yellow or orange. If there's no color change or no precipitate, then that means there's no sugar. So it's kind of a qualitative measurement here, but it's pretty obvious whether or not there's sugar. But let's say that we did this test and we tested the dogs with an intact pancreas and the dogs that were missing their pancreas, and we got no precipitate, no color change. We might conclude that, okay, the pancreas has nothing to do with regulating sugar. And, well, we know now that would be wrong. So what we want to do is we want to test the Benedict's reagent. So what we could do is we could do a positive control where we take some urine from a healthy dog and we add sugar to it. So on purpose, we're going to add some sugar, mix it up, and then we add some Benedict's reagent and make sure that the Benedict's reagent actually changes color. I mean, it's possible there's something in the urine that could stop it from changing color. So if we did our test and everything came out the same, then we would go back and we would do this positive control. And in this case, the positive control is just to test that the Benedict's reagent is working and it's okay. And that's the most common use of a positive control. Okay, so looking at this experiment again, so our question, of course, was, does the pancreas play a role in regulating sugar levels in urine? Our experiment, we have our independent variable, and that's just whether or not the pancreas is there. We have our dependent variable, that is whether or not there's sugar in the urine. We have our experimental group, that would be the dogs with the pancreas removed. We have the negative control, that would be dogs that have not had their pancreas removed. They've undergone the surgery, most of it, but they haven't had the pancreas actually removed. The positive control would be to take some normal urine, add some sugar to it, mix it in, test it with the Benedict's reagent, just to make sure that that reagent is working. Our hypothesis, of course, is that the pancreas makes some sort of factor. We know it's insulin now, they didn't know that, that will limit the amount of sugar that gets into the urine. And the prediction, based on that hypothesis, is that dogs without a pancreas will have sugar in their urine. And healthy dogs that have a pancreas will not, or at least they'll have a lot less. So this is the common scientific method. This is a methodology that we use when we can do experiments. But the problem is we can't always do experiments. And you might actually be surprised to learn that very often we can't do experiments. A lot of science is observational. And that means we look at stuff and we take down lots and lots of data and we try to make rational conclusions based on that. But we can't always do an experiment. If we can do an experiment, that's great. But what if we're a geologist? Can you do an experiment to explain geology? Not really, just don't have that much time. So regardless of how data is collected or generated, science relies on deductive reasoning. We come up with hypotheses, and then we go out into the world, and sometimes we test them with actual tests, with experiments, but sometimes we test them just by making observations. And we look for things that can falsify our hypothesis. So we try to look for the things that we know will prove us wrong. And if you're a really good scientist, that's what you're doing. You're looking for things you know will prove you wrong. And every time you fail to find them, that offers some support to your hypothesis. So I mentioned that we're somewhat limited when we're doing things like geology, for instance, when it comes to experiments. There's some experiments we can do, but let's say we want to do an experiment on how volcanoes form. Well, volcanoes form over millions of years. We can't really recreate that. What we can do, though, is we can still come up with a hypothesis, and we can go out there and test it by making observations. And we're looking for stuff specifically that will prove us wrong. So if you're a good scientist, you're always looking for data or observations or experimental evidence that will prove you wrong. And every time you fail to find that, then that offers support to your hypothesis. Let's look at the example of the extinction of the dinosaurs. So the dinosaurs went extinct 65 million years ago. And it's thought that there was a giant asteroid that collided with the Earth. In fact, it's thought that it carved out the Gulf of Mexico. That's where this impact is thought to have taken place. And what that did is it threw up a whole lot of dust and ash and smoke and so on into the atmosphere. And for several years after that, the skies were darkened, plants died, dinosaurs died, etc. We had this mass extinction. We can't recreate that in an experiment, but our best evidence for this is that all around the world, if we look at sediments, so let's say we have a layer of rock. As we go deeper and deeper, the rocks get older. They were laid down earlier. And when we get back to the rock that was laid down 65 million years ago, all around the planet, there's a layer of clay and ash that contains something known as iridium. Iridium is an element that's fairly common in the universe, but not that common on Earth. Most of the iridium that we see on the Earth comes from meteorites. So this layer of clay and ash is thought to be the remnants of the meteorite. It smashed into the Earth, it threw up all of this dust with all of this iridium in it, and that settled out. Now, we find dinosaur fossils lower down. We find dinosaur fossils deeper down in the rock record, but we've never found dinosaurs above. So if we want to test our hypothesis, we can just go out there and look for dinosaur fossils that are younger than that iridium layer. We can look for dinosaur fossils that are younger than 65 million years and are above this layer that most probably came from a massive asteroid impact. And every time we fail to find those dinosaurs, we offer support for our hypothesis. So even though we're not doing an experiment, we're still following the same methodology. A lot of science is not experimental. It's observational. And in fact, you might be surprised that most science is observational. This is sometimes referred to as discovery science. This is where you're going out there and you're just making observations. So as examples, if we're looking at tissues, if we're looking at sections of human tissue and we're looking at the structure of neurons and stuff like that, we're not doing experiments. We're just describing the structure of the neuron. Maybe you're out there catching insects and you find a new species and you're just describing that new species. And that is a lot of science. A lot of science is just making observations, trying to make sense of them. If you're trying to identify stuff, you're categorizing it. You're just keeping everything kind of neat and tidy and making clear, concise, logical observations. And finally, I want to point out that science is always looking for natural explanations. Ideally, we're looking for something we can measure, but definitely it has to be something that's falsifiable, something that's testable. So we can't necessarily test supernatural explanations. You can't really use science, it depends on your point of view, to suggest that God doesn't exist, for instance, because God is maybe not testable. And if it's not testable, it's outside the realm of science. So there are, of course, limitations to what we can test. And the big one is that it has to be falsifiable. It's really difficult, if not impossible, to use science, or logic for that matter, to show the non-existence of something. I could say that I don't think aliens exist. And I could spend every night looking through a telescope and looking up at the sky, and my whole life I don't see an alien. I don't see a UFO. Does that mean they don't exist? No, not necessarily. You cannot prove the non-existence of something. And that's a limitation of science, but like I say, it's a limitation of rational thought as well. So I don't want this to seem like a diss against religion. I'm just saying that religion and other supernatural experiences and feelings and so on are really outside of what science can help us with. And finally, I have a few more terms to throw at you. Terms that unfortunately are thrown around all the time and have very different meanings in science than they do in everyday speech. So what is a fact, for instance? A fact is something that most people would agree is kind of non-negotiable. It's proven to be. And a good example would be, let's say I said, Paris is the capital of France. Well, we know it's the capital of France. France decided it's their capital. You can look in any atlas, you can talk to any French person, they'll tell you that Paris is the capital of France. That's a fact. When it comes to science, it gets a bit trickier. If we have enough evidence, then we might say it's a fact, but it depends who you talk to. Some people want more evidence than others. I'm a person that wants lots of evidence. And remember, when we're doing science, we're trying to disprove things. We do the experiments or the observations over and over again, and every time we fail to disprove something, we've offered support that it's correct. But how many times do we have to do that before we can say it's a fact? That's open for debate. What is a law? Now, a lot of people think a law is something that's proven true, and that's kind of true, but when we're talking about science, a law is something that's quite small. Laws are usually ratios. You might have heard of the gas laws. You might have heard of the law of universal gravitation. Those are laws. Those are simple mathematical ratios typically. A theory, that word means something very, very different in science than it does in everyday speaking. If you tell your friend that you have a theory, usually you mean that you have a hunch, and it means something very different in science. A theory is a model. A theory is a collection of facts, a collection of laws. It's a really big thing with lots of supporting evidence. So we have the cell theory. The cell theory tells us that living things are made up of cells, and I don't think anyone would debate that. We have the theory of evolution. Again, saying it's a theory doesn't mean it's a hunch. It means it's a model, and it's supported by lots and lots of evidence. What we're seeing here, this is Banting and Best. They were researchers at the University of Toronto, and they isolated insulin from the pancreas. So they were the ones that actually showed that, yes, the pancreas regulates sugar levels. They used dogs, unfortunate dogs, for their experiments as well, but they were able to isolate the compound that's found in the pancreas that reduces sugar levels in the blood and urine. And this is their model, their theory of how this works. And it's supported by tons of evidence by lots of different researchers. So insulin stimulates the cells in your body to take up excess glucose, excess sugar, and that reduces the amount of sugar in the blood, and that reduces the amount of sugar that gets into the urine. But again, a theory is not a hunch. A theory is a model that has lots of bits and pieces to it. So to sum up, at the end of each of my PowerPoints, I will have some concepts, and these are just really broad concepts that we've talked about in the lecture. The universe has this hierarchical structure to it that we talked about. Biology has a hierarchy to it. Life is difficult to define. We talked about some of the characteristics you might use. There's others you could use as well. And the cell is the basic unit of life. So every living thing is one cell or a collection of cells. Individuals that have traits that allow them to survive and find mates are more likely to pass those traits along to the next generation, and that's the essence of evolution. That's how evolution works. For science, it's the process of logically and systematically testing a hypothesis, and remember, we're always trying to prove it wrong. You have your hypothesis, you come up with an experiment that you don't think is going to support it, you come up with an experiment that you think is going to prove it wrong. And then you do your experiment, and if it doesn't prove it wrong, then bravo, you've had a bit of support for your hypothesis. And then you go and design a different experiment that could possibly prove it wrong in a different way. Hypothesis has to be testable and falsifiable. So again, if I say that there's aliens on the other side of the galaxy, I can't test that. I can't falsify that. That is a useless hypothesis. We can't test all hypotheses, so we have to start with the simplest, and if that doesn't work, then we can get more complicated. And for each of my PowerPoints, I will have a terminology list. These are the terms you should focus on. So when you're studying, focus on these terms. These are the terms I'll use in my test questions, for matching questions, for true, false, etc. So this is the key points that I want you to know. And a few study questions thrown in as well. See you next time.